Hello, Dr. Manella. This is my eReport for the molecular biology portion of this course. The first topic I'm going to discuss is DNA compaction. The first step in DNA compaction is to coil the protein-free DNA around a nucleosome. A nucleosome is made up of 4 histone proteins: H2A, H2B, H3, and H4. 2 of each of these proteins comes together to form an octamer. This octamer acts as a histone spool that the DNA can coil itself around. The histones carry a positive charge and the DNA carries a negative charge. This allows the two to bind securely to each other. About 147 bases wrap around the histone before exiting the spool. A protein-free strand of DNA exits, and only until about 5 to 200 bases later does the DNA begin to wrap around another histone spool. This span of protein-free DNA is known as linker DNA. This coiling around the nucleosomes is the first level of DNA compaction. The next stage of DNA compaction utilizes the H1 histone protein. H1, which can be seen here, interacts with H4 in the octamer in order to position itself perfectly on the DNA right as it exits the nucleosome. H1 then kinks the DNA to promote a twisting structure. The DNA is then able to coil itself. Here you can see the top view of that coil, and over here you can see this is the side view of that coiling. The nucleosomes are getting much closer together in a twisting motion. This is known as the 30 nanometer fiber. The next stages of compaction are not very well known, but the 30 nanometer fiber will become 10,000 times more condensed in order to form the mitotic chromosome. The one catch to DNA compaction is that it must be reversible, because every gene sequence must be available at a moment's notice. And this can be achieved through localized unwinding, with the use of 2 specific enzymes, which are known as HATS and HDACs. The histone proteins themselves in the octamer have tails that are very rich in the amino acid lysine, which carries a positive charge. That positive charge is why DNA can bind so securely to the histone spool. The first enzyme is histone acetyltransferases, also known as HATs. HATs acetylate the lysines on the tails of the histones. This neutralizes the positive charge on the lysines. Without the positive charge, the DNA is no longer strongly attracted to the histone. This will actually loosen the DNA and making it more accessible for transcription. And this is how we can access genes at a moment's notice. If they're wrapped around the histone spool, they're not easily accessible. But once they're loosened, these can then be transcribed. The other enzyme is histone deacetylation complexes, which is the HDACs. HDACs deacetylate the lysines on the histone tails, which restores their positive charge. This increases the interaction between the DNA and the histone, locking that DNA into place, preventing access to those gene sequences. HATs and HDACs work together to make genes accessible and genes inaccessible when needed, and help control gene expression by controlling the accessibility of DNA. Cells are dividing constantly, which means that DNA replication needs to occur just as often. The base pairing system enables replication. This means A always pairs with T and G always pairs with C. When the 2 strands of DNA are pulled apart, each strand acts as a template, allowing for the new complementary strand of DNA to be made off of it. Once replication is complete, the DNA consists of 1 old strand and 1 new strand of DNA. This is known as semi-conservative replication. For replication to begin, the 2 strands of DNA must be separated. There are countless hydrogen bonds that tightly hold the 2 strands together. At certain sequences in the DNA, there are origins of replication. Initiator proteins bind there and force the two strands apart by breaking those hydrogen bonds. This causes the formation of the replication bubble. The replication bubble can be seen in the upper right-hand side of this diagram. The replication machinery attaches to either side of the replication bubble. The active site in this machinery is called the replication fork. The two replication forks, once attached, move in opposite directions down the strands of DNA. One of the most important enzymes in the replication fork is DNA polymerase, as it synthesizes the new strand of DNA. DNA polymerase is able to make the phosphodiester bond between the 5' phosphate of the incoming nucleotide and the 3' hydroxyl of the previously placed nucleotide. When the nucleotides enter the reaction site of DNA polymerase, they are called deoxynucleotide triphosphates, or DNTPs. When the two phosphates on the DNTP are cleaved, it releases a massive amount of energy that allows for replication to proceed. If DNA polymerase were left to its own devices, it would leave the DNA strand after placing only a single nucleotide. There are two proteins that are used to prevent it from leaving. Those are the clamp protein and the clamp loader protein. The clamp protein holds the DNA polymerase in place, and with the energy from ATP, the clamp loader protein opens the clamp protein and threads the DNA into the clamp. This holds DNA polymerase onto the DNA strand, which allows it to place one nucleotide after the other, all the way until DNA replication is complete. There are many proteins and enzymes that play a crucial role in DNA replication. Two of these are DNA helicase and single-stranded binding protein. DNA helicase spins rapidly to pull the two strands of DNA apart after the replication bubble has formed. The single-stranded binding protein binds the now single strands of DNA that have just been pulled apart by DNA helicase to ensure that they remain apart and do not reassociate with its complementary strand. These two allow the replication fork to continue moving down the DNA strand for replication. The replication fork itself is asymmetrical due to the fact that DNA is synthesized in a 5' to 3' direction. There is a leading strand, which is the top strand in this diagram, that synthesizes DNA continuously. This is because it moves towards the replication fork as it pries apart the two strands of DNA ahead. But there is also a lagging strand. This is where the DNA must be synthesized discontinuously because it moves away from the replication fork. This causes the new strand of DNA to be made in segments. These segments are known as Okazaki fragments and must later be joined together to make one continuous strand of DNA. The one problem with DNA polymerase is that it's only able to add a nucleotide to a 3' hydroxyl group. This means that it cannot synthesize DNA from scratch. This is where primase, a type of RNA polymerase, comes in. Primase is able to make RNA from scratch, unlike DNA polymerase. Primase lays down about 10 RNA bases, forming the RNA primer. Because DNA and RNA are so similar in structure molecularly, DNA polymerase can add new nucleotides to the RNA primer. The leading strand of DNA only requires one RNA primer, while the lagging strand needs a new RNA primer for every Okazaki fragment that is made. Because the lagging strand needs an RNA primer for every single Okazaki fragment, it is imperative that the lagging strand not be left as this RNA-DNA hybrid. And there are a few steps to fix this. An enzyme known as repair polymerase chews away the RNA primer from the 3' end. The same enzyme is able to lay down DNA bases in place of the primer. Finally, DNA ligase joins the phosphate sugar backbones of the newly added nucleotides to create one continuous strand of DNA, and not an RNA-DNA hybrid. The final process of DNA replication is the addition of telomeres. Unfortunately, primase is unable to place the RNA primer in front of itself, which means that it needs a place to land. And without a solution, each round of replication would result in a shortening of the DNA. This is because RNA is chemically unstable and it degrades quickly, leaving gaps at the end of the chromosome, as seen in this upper strand of this diagram. This is known as the end replication problem. The solution to this problem? Telomeres. Telomeres are caps on the chromosomes that do not rely on replication. They provide a place for primates to bind without causing a shortening of DNA. These telomeres are produced by an enzyme called telomerase. Telomerase is attracted to the ends of chromosomes, and it carries a short template strand of RNA. Telomerase is also able to act as a DNA polymerase, so it's able to make DNA that's complementary to the piece of RNA that it's carrying, as seen here. This RNA template allows telomerase to make short DNA sequences over and over again in tandem until the cap of DNA is made at the end of the chromosome. This repetitive process creates an extension of the DNA sequence, as seen here. Primates can then bind to this extension that's been formed by the telomerase and leave an RNA primer. Then DNA polymerase comes and finishes base pairing the sequence. Though this doesn't solve the end of replication problem, it shifts it though. It goes from a gap in important chromosomal DNA to a gap in repetitive sequences that are in tandem that the telomerase carries around. It acts as a buffer to prevent the loss of important coding regions of DNA. Once the telomeres are made, DNA replication is now complete. The DNA mismatch repair system is used to fix bases that have not been paired correctly in DNA replication. This system fixes 99% of mistakes and detects them by feeling for a distortion in the DNA, as seen here with this GT mismatch. The one problem with the mismatch repair system is that before repairing the mismatch, the cell must determine which strand of DNA is reliable, because if the cell fixes the wrong base, the mutation will be permanently fixed into the genome. In bacterial cells, the reliable old DNA strand is methylated, as denoted here in the diagram. The strand that is not methylated will be the strand that is altered using the mismatch system. And it's actually unknown how eukaryotic cells accomplish this, because methylation in eukaryotic cells is used for epigenetics, and not for the mismatch repair system. Once the bacterial cell has determined which base needs to be excised, using methylation, the cell then nicks the backbone of the DNA strand at the methylation and chews up the DNA all the way up to the mismatch. Repair DNA polymerase then comes in and places nucleotides in where the strand has just been chewed away. Finally, DNA ligase will come in and seal the phosphate sugar backbone, creating a continuous strand of DNA with the fixed mismatch. This system is actually our best defense against somatic mutations and cancer. The cell also has systems in place to repair a double-stranded break. Double-stranded breaks in DNA are a complete severing of a DNA molecule into two pieces, and both sections are broken in the same place. Radiation can be a cause of these double-stranded breaks. This is potentially a life-ending mutation for a cell. And because the DNA is cut in half, there is no template strand to serve as a guide for repair. Cells have two mechanisms in place to help repair double-stranded breaks. The first is non-homologous recombination. Unfortunately, non-homologous recombination is not the most ideal way of fixing double-stranded breaks, but it's a lot better than the cell dying. The broken ends are blunted and then ligated back together. Unfortunately, during this process, nucleotides are lost, so the cell is basically crossing its fingers and hoping that this isn't a protein coding region of the DNA. Homologous recombination is the preferred method for fixing double-stranded breaks, but it's not always feasible. Homologous recombination can only occur if homologous chromosomes are near each other in the nucleus. This is because the homologous chromosome will serve as a template for the repair. Though the sequences might not be identical, but it's the best we've got. It's better than the cell dying, and it's better than losing nucleotides in a possibly important protein coding region of DNA. To begin homologous recombination, a nuclease removes nucleotides from one strand on each side of the break. So here nucleotides were removed, and here nucleotides were removed. Removing these nucleotides creates a 3' overhang, which are these two extensions of DNA. If the homologous chromosomes are near each other in the nucleus, this bottom 3' overhang will invade the double-stranded DNA and will search for a sufficiently similar sequence that it can base pair with. Once a sufficiently similar sequence is found, this 3' overhang will actually base pair with this bottom strand of DNA. This will displace the upper strand of DNA, causing the formation of a D-loop. This also forms something known as a branch point. This is where the two strands of DNA cross over. Once the invading strand has sufficiently base paired, DNA polymerase will elongate the invading strand by using the intact strand as a guide. As this elongation occurs, the D-loop will actually extend as well. As the D-loop elongates, this will actually align it with the top 3' overhang. The top 3' overhang can now base pair with this. DNA polymerase will come in and start lying down nucleotides that match the intact strand. When homologous recombination is complete, the two strands now must be detangled from each other. These two branch points, these 2 points of crossover, are nicked by DNA ligase. This allows the strands to untangle. DNA ligase also seals the phosphate backbones together once again. This will leave two completely intact molecules of DNA, repairing that double-stranded break. This is the preferred mechanism over non-homologous recombination because no nucleotides are going to be lost in this process. Initiating transcription in eukaryotic cells is much more closely regulated and complex. Because of this, it requires many proteins. RNA polymerase II is what forms the mRNA strand in eukaryotic cells, but it is unable to initiate transcription on its own and requires general transcription factors to begin the process. General transcription factors assemble at the promoter with RNA polymerase II, forming what's known as the pre-initiation complex. All of the proteins in the pre-initiation complex work together to make sure that transcription happens and it happens smoothly. Generalized transcription factors are responsible for recruiting RNA polymerase II to the promoter and allowing it to move down the gene. The promoter in eukaryotic cells is known as the Ta-Ta box. It's called this because of its high concentration in thymines and adenines. The high concentration in A's and T's actually makes the DNA strands easier to separate, due to only 2 hydrogen bonds lying between the AT base pair, instead of 3 hydrogen bonds between the GC base pair. The first general transcription factor that's able to recognize and bind to the promoter is TF2D. It's able to do this because it has a subunit known as the Ta-Ta binding protein, or TBP. TBP is the only one that's able to directly bind to the promoter. It actually kinks the DNA at a 90-degree angle to inform the cell that, yes, this is definitely a promoter. The next general transcription factor to bind is TF2A. Once this binds, TF2B binds next. The next general transcription factor to bind is TF2F. TF2F is what brings RNA polymerase II to the promoter. Once TF2F and RNA polymerase II is there, the next general transcription factor to bind is TF2E. And finally, the last general transcription factor to bind is TF2H. And this completes the pre-initiation complex. RNA polymerase II, much like the histone proteins discussed at the beginning, have a tail. This tail is a chain of amino acids that are accessible and modifiable. The final generalized transcription factor to bind was TF2H, and this actually has a major influence over RNA polymerase II because it's able to phosphorylate the tail of RNA polymerase II. If the tail of RNA polymerase II is phosphorylated, then RNA polymerase II is able to move down the DNA strand. Aside from phosphorylating the tail, TF2H also pulls apart the DNA strands while RNA polymerase II is moving down the DNA. Once RNA polymerase II is successfully transcribing the gene, A large majority of the generalized transcription factors dissociate from the promoter. Once RNA polymerase II passes a stop sequence, the tail will get unphosphorylated, which allows it to fall off the gene. This is important because only an unphosphorylated RNA polymerase II is able to be recruited to the promoter. After transcription, a pre-mRNA molecule is formed, and it needs to be processed before it is allowed to leave the nucleus. It must get capped, polyadenylated, and spliced before exiting. First, the 5' end of the mRNA gets capped with an inverted methylated guanine, with all 3 of its phosphate groups intact. This marks the beginning of the mRNA sequence as genuine. Then polyadenylation occurs. This is when the 3' end of the mRNA receives a poly-A tail. This is a run of hundreds of adenine bases, which marks this end of the mRNA as genuine. Once the beginning and the end of the mRNA is marked as genuine, the final step in mRNA processing is splicing. Almost all of our protein-coding genes are interrupted by long non-coding sequences known as introns. The actual protein-coding regions of a gene are known as exons. These introns can vary from 1 to 10,000 nucleotides long and must be spliced out of the mRNA sequence before it can leave the nucleus. Introns must contain 3 identical sequences that identifies it as a spliceable intron. 1 sequence is at the beginning, 1 is at the end, and finally a middle section that contains 1 adenine. A spliceosome then uses these sequences as a guide for removing introns, then subsequently ligating the exons together. The spliceosome breaks the 5' end of the intron, freeing it. The now freed 5' end of the intron attaches itself to the adenine in the middle section of the intron. This forms a lasso-shaped structure known as a lariat. The lariat gets cut at the 3' end, which removes the intron completely from the mRNA sequence. The remaining exons then get ligated together. Once the introns are removed and the exons are ligated together, the mRNA is now fully processed and can exit the nucleus. Though the functions of introns are not well known, they are known to have one purpose, and that is alternative splicing. Alternative splicing allows for some exons to be deleted, which allows non-adjacent exons to be ligated together. This creates slightly different mRNA sequences, which allows for the formation of a variety of different proteins with slightly different functions to be made from the same gene. It is believed that 60% of human genes can undergo alternative splicing. This greatly increases our protein potential. If a specific exon encodes for one part of the protein's function and that exon is spliced, that one function of that protein is no longer there. If an exon codes for ATPase, which is an enzyme that allows for ATP to be broken into ADP, if that exon is spliced out through alternative splicing, that protein can no longer act as an ATPase, yet its other functions remain intact. This greatly increases our protein potential because we can modulate the protein's functions through the use of the same gene. The translation of mRNA to protein is done through codons. Codons are a set of 3 nucleotides that equates to a single amino acid. The tRNAs are the bridge between the language of nucleotides and the language of amino acids. tRNAs have an anticodon on one end, which is able to bind to and directly pair with its corresponding codon. And it is loaded with the proper amino acid on the other end by aminoacyl tRNA synthetases. Ribosomes help coordinate this process of translation. The ribosome itself is made up of 2 parts: the smaller subunit, which binds to the mRNA strand and coordinates the proper matching of the codons and anticodons, and the larger subunit, which builds the amino acid chain by forming the peptide bonds between each amino acid. When these 2 subunits come together, they trap the 5' end of the mRNA between them. The large subunit has 3 sites in it that accommodate the tRNA. These are the A site, the P site, and the E site. The A site accommodates the tRNA known as aminoacyl tRNA, which is a tRNA carrying only one single amino acid. The P site accommodates the tRNA carrying the peptide chain, also known as peptidyl tRNA. And finally, the E site. This site is for exiting tRNAs that are no longer carrying any amino acids. The first step in translation is initiation, which is not an easy feat to accomplish. Eukaryotic initiation factors, also known as EIFs, are responsible for initiating translation. EIFs bind to the small subunit of the ribosome and the 5' cap of the mRNA. They help by positioning and sequentially recruiting all of the necessary players for translation. Without EIFs, translation cannot occur. The problem with tRNA initiation is that a tRNA carrying only one amino acid must enter the P site and not the A site. And as stated before, the A site is what accommodates a tRNA with a single amino acid on it. The only tRNA that is able to accomplish this by breaking the rules is the initiator tRNA. The initiator tRNA carries the anticodon for the AUG start codon. It also carries the methionine amino acid, but the methionine has a formal group bound to it. This is called FMET. FMET is only carried by the initiator tRNA. When the smaller subunit binds to the 5' cap of the mRNA, it engages the initiator tRNA while it scans for a start codon. Once found, the initiator tRNA binds to the start codon. The larger subunit then binds to the smaller subunit, ensuring that the initiator tRNA is in the P site. Also, the EIFs help to make sure that FMET lands in the P site and not the A site. The next step that occurs in translation is called elongation. The elongation of the amino acid chain occurs in 4 repetitive steps. First, the tRNA with the correct anticodon that is carrying the appropriate amino acid enters into the A site and base pairs to the codon. Secondly, the amino acid on this tRNA forms a peptide bond with the amino acid chain that is linked to the tRNA in the P site. This is shown through this red arrow. This then transfers the entire amino acid chain to the tRNA in the A site. Third, the ribosome then shifts forward by 1 codon in the 3' direction, while the tRNAs are still base paired with their codon. This is a process known as translocation. Translocation moves the now empty tRNA in the P site into the E site, and the tRNA in the A site that is now carrying the entire amino acid chain moves into the P site. Finally, because only 2 tRNAs are able to be in the ribosome at any time, when a new tRNA enters into the A site, the tRNA in the E site now exits. The mRNA is pulled through the ribosome as this process repeats over and over and over again until a stop codon is reached, which then triggers termination. The final step in translation is known as termination. When a stop codon enters into the A site, proteins known as release factors enter into the A site. This is because there are no tRNAs that are able to recognize the stop codons. These release factors catalyze the release of the amino acid chain from the tRNA in the P site. Once this occurs, the polypeptide chain is now free and can float away from the ribosome and begin to fold into its natural 3D shape. After this, the large subunit will try to translocate, but the smaller subunit cannot move. This causes the 2 subunits to dissociate, releasing the mRNA from the ribosome. These ribosomal subunits and mRNA can now be used in translation once again. Life inside a eukaryotic cell is complex, and each pathway is highly dependent on other pathways in the cell. This requires a high level of refined gene regulation. A single eukaryotic gene must be regulated by a combination of many different proteins for this very reason. This is the basis of combinatorial control in the use of the mediator protein. Eukaryotic transcription factors are able to bind to enhancers, which can lie thousands of bases away upstream or downstream of the target gene. This means that regulatory sequences for one gene are scattered throughout long stretches of the entire chromosome. These enhancers and transcription factors are able to interact with proteins on the promoter of the target gene, even though they can lie thousands of bases away. The DNA in between can bend and manipulate itself in order to allow the enhancers and the regulatory transcription factors to be able to interact with these proteins. There are very few genes in our cells that are regulated by just one transcription factor. Typically, the genes in our cells are turned on and off by a committee of proteins. Combinatorial control is when groups of regulatory proteins, each bound to their individual enhancers, are working together to precisely regulate and control the expression of a gene. Dozens of these regulatory proteins, activators and repressors, each add their own unique signal depending on the conditions in the cell or outside of the cell. Each regulatory protein adds information that will help determine whether or not RNA polymerase II will leave the promoter. It all depends on which signal tips the scales in favor of one direction or the other. One regulatory protein could be influenced by glucose levels in the cell, and another may monitor if there are other types of sugars available to the cell in the extracellular environment. There is a massive protein complex that's made of about 20 different proteins that sits on the end of RNA polymerase II. This is known as the mediator complex. This complex is what counts the votes from all of these transcription factors and other regulatory proteins. The mediator takes in all of the different signals that affect a single gene and integrates that into one cumulative signal and relays that final signal to RNA polymerase II. This could either be a signal to leave the promoter and begin transcribing that gene, or a signal to repress that gene and halt transcription. It all depends on how the scale was tipped. RNA interference, also known as RNAi, is a form of post-translational regulation and is believed to be originally used as a defense against viruses. This mechanism is used for gene silencing and is only found in eukaryotic cells. It is also known as the most potent form of regulation. When double-stranded RNA enters the cell, it triggers the RNAi process, but it can only do that if the double-stranded RNA is 21 to 23 base pairs long. If the double-stranded RNA is too long, an enzyme called dicer shreds it into the proper length. These fragmented strands are known as small interfering RNA, siRNA. When siRNA is formed, one of the two strands then binds to the RNA-inducing silencing complex, known as RISC. Using the siRNA, RISC can now seek out any complementary sequences and destroy them. If this mRNA sequence is ever produced by the cell, once it exits the nucleus, it will be targeted for destruction and that protein will never be produced. This is known as the most potent form of regulation because about 50 siRNAs are able to silence thousands of pre-existing mRNA molecules through the use of amplification. This amplification is possible through the use of RNA-dependent RNA polymerase, RDRP. Once RISC cuts the mRNA molecules into unstable fragments, RDRP uses one of the fragments as a template to generate more double-stranded RNA, which is then shredded by dicer, which generates more siRNAs for RISC to bind to. And it does this over and over and over again, allowing RNAi to be able to instantly halt that protein synthesis. That cell will never be able to produce that protein ever again due to RNA interference.